Back to the cephalic phase. So this is initiated by taste receptors, olfactory receptors, the thought or sight of food, so vision, sight. This is going to be triggering the, so it's okay, senses or thought, triggering sensory receptors, ultimately triggering the cerebral cortex. Usually you're aware that you've smelled or tasted or thinking of food, hypothalamus. And then we're going to have the vagus nerve, which is parasympathetic, also some medulla, parasympathetic nervous response to the cells of the stomach. Okay, so let's tie that in, cells of the stomach. What cells are going to respond here? We're going to have, indicate this here, mucus cells, chief cells, parietal cells, and we'll just edit here, okay, G cells. These are all going to produce their things. I'm going to do this in a different color here. So we're going to have mucus. We're going to have pepsinogen. We're going to have HCl. And we're going to have gastrin. You know what all of these do? These are all a critical part of digestion, except for gastrin. What does gastrin do? It's actually going to feed back and increase both of these, almost a little positive feedback thing there. This here is a paracrine. It's acting as a paracrine from this enteroendocrine cell, which is a G cell. So this results in a huge amount of increase in gastric secretions, about 500 milliliters per hour. Remember I told you normally it's about 3 milliliters per day. Is that right? But it's very short, so it doesn't last an hour. So it's just a high, quick rate. Lots of acid. You're also going to, no, that's it. Okay, the next phase is if you actually eat, right? This could happen, the cephalic phase, and you smell food and you know how it makes you hungry, and you actually do have increased gastric secretions, but you didn't actually eat anything. If it enters your stomach, the gastric phase begins. So this is when food enters stomach. What is that food? What kind of stimuli does it contain? It's going to have stretch, result in stretch. It's going to increase the pH of your stomach. Anything you ingest, pretty much, even orange juice and coffee, which are acidic, are still going to increase the pH of your stomach. Your pH of your stomach is super low. That's a stimulus. There's going to be other chemicals in food that can be detected. So these are detected by stretch and chemoreceptors. And there's going to result in a neural response and a hormonal endocrine response. So the neural response is detected by enteric neurons. Remember the enteric nervous system? These actually secrete additional acetylcholine. What else secretes? Why did I say additional? Well, the vagus nerve is secreting acetylcholine. Yeah, so more acetylcholine is going to be stimulated locally here. This is a local neural response that's going to augment and increase the effects of the vagus nerve that already stimulate this from the cephalic phase. So this is going to result in all those 4 cell types we talked about before, right? Those are going to be stimulated even more. That was the result of the vagus nerve stimulation. Mucus cells, chief cells, parietal cells, G cells. So it's going to result in even more HCl, pepsinogen, gastrin. This gastrin. This is part of hormone response. The other thing that triggers gastrin, besides the local nervous response, is partially digested peptides specifically. That's one of the chemicals in the food that is going to stimulate our G cells to stimulate gastrin release. So that's part of that specific hormone response. The other things that's going to happen besides gastric secretions, literally increased gastric juices, is going to be from both of these. Let's see, from both the enteric neurons directly and gastrin are both going to increase peristalsis, which is going to increase mixing, right? Like stomach activity, your stomach mechanically digesting your food. And now we're actually, right, these things are all actively digesting food. And as we have, as pepsinogen and HCl do their thing, that's going to cause our proteins to be partially digested and cause increased mixing. So it's a kind of a positive feedback system. The last phase is the intestinal phase. This is when food enters the intestine. And this is where we want to be able to regulate gastric secretions. So to regulate gastric activity, gastric juice secretion, and like peristalsis and movement, we want to be able to do this so that we can control what's coming in right here. We want to be able to, if food is not digested, if too much is coming in, we want to be able to regulate that. This is the last phase of gastric regulation. After several hours of mixing in the stomach, chyme starts to enter the duodenum. And we want to be able to control the rate at which that happens. So the small intestine can effectively do its job in digesting and absorption. We don't want chyme to be flowing in like too fast. So as chyme enters the duodenum, we've got, guess what, stretch and chemoreceptors in the duodenum that are going to respond. They're going to trigger the enterogastric reflex, a reflex that both feeds back a short reflex to the enteric neurons of the stomach, and there's a long reflex to the CNS to, what do you think, decrease parasympathetic, increase sympathetic. And that's then going to go to the stomach. These are both, what was I gonna write? This is our short reflex and our long, which you already know those terms, right? Both of these reflexes are going to, let me do a different color, inhibit gastrin release, inhibit gastric activity, close the pyloric sphincter, shut off digestion coming into the intestine. The last thing is the hormones produced in this small intestine here. When you've got arrival of lipids and carbohydrates, you have in the small intestine, that's going to result in CCK and secretin release. Hormones produced by the duodenum itself. So lipids and carbohydrates had stimulated CCK and secretin. Another thing that can stimulate these two hormones is a decrease in pH. So as chyme enters the duodenum, that's going to decrease pH within the duodenum. This isn't related to stretch receptors actually. So let me actually do that here. The chyme is going to stretch stuff. It's also going to have a low pH. That low pH also triggers CCK and secretin release. I haven't told you what these things do yet, but maybe you can guess, right? If we have a low pH in the chyme, what are these going to do to gastric activity? We want them to turn it off, slow it down. They're going to inhibit, I don't have those cells written on this slide, the chief cells, the parietal cells, I believe is actually mostly those two, chief and parietal, which is our pepsinogen and our HCl. Those are going to decrease. We don't want to make more acid, right? If we're already having a lot of acidic acid come from the chyme, we're maintaining that variable relatively well. So that's negative. We also have a negative effect on peristalsis. We're going to inhibit peristalsis, which is going to decrease motility of the stomach and decrease what's coming out into that small intestine. So this whole intestinal phase, regulate gastric activity, typically decrease. There are exceptions to that in terms of specific stimuli. So for example, partially digested proteins will feed back to increase gastrin release to speed gastric processing because the proteins should have been digested in the stomach. Okay, we'll come back to CCK and secretin when we get to the small intestine because they are also going to regulate and stimulate the pancreas and bile ejection. Okay, I want to review for you with a different diagram here because this is a little messy, which is fine, right? Right, totally fine. I want to do this one more time here with a nice summary. So we've got intestinal phase, presence of fatty, hypertonic, so like high osmolarity, really salty, acidic chyme. Where? In the duodenum. I think some people say duodenum. I don't care. We're going to have a endocrine response, hormone response, and a neural response. Let's do neural response first. Chemoreceptors and stretch receptors are going to detect the stimulus, potentially, high protein as well, but let's not do that one. This is going to trigger our enterogastric reflex initiated in the small intestine. And there's both a short, actually, I'll do that over here, a short to the enteric neurons of the stomach and a long via the central nervous system, increase sympathetic, decrease parasympathetic, because we want to decrease digestion, right? We want to decrease, and that's the last step here. These things are going to decrease peristalsis, stomach motility, They are going to decrease, don't have any room down there, decrease emptying right into the duodenum, also going to decrease gastric juices. The hormonal response is detected by these, actually, let me see this. There we go. Yeah, by the duodenal endocrine cells. They're actually also called enteroendocrine cells because they're part of the digestive system, but whatever. The endocrine cells themselves, right, can act as stimulant receptors, just like the pancreas can, remember? They are going to secrete these hormones, secretin, CCK, there are others, hormone release. They're hormones because they're traveling in the bloodstream. They're going to have the same effect by acting on the stomach itself. They're also going to travel through the bloodstream and act on our pancreas and gallbladder.